Okay, so simian virus for multiplication cycle, or what is this? This is Polyomaviridae, so it's a tumor. It's a tumor virus. Now, tumor viruses are double-stranded DNA, or they have double-stranded DNA genome, and because of that, they actually use the same histomolecules that our cells would use, right? Because you need to wrap the double-stranded DNA, you need to condense it somehow, and what better way to condense it than to just jack the histones that were already made in the cell. So that's why these virion particles that you see right here come with H2A, H2B, H3A, H4. Those are histone proteins that the double-stranded DNA wraps around. And then you also have these capsid proteins. VP1 is functions in structure and attachment. VP2 and VP3 function in structure and encoding. The majority of capsid is made up of VP1. The minority of the capsid is made of VP2 and VP3. So the virion particle adsorbs to the MHC class 1 cell receptor onto the cell. Adsorbs just basically means that it's held as a thin-like film or structure on the outside of the cell. Essentially, the way I see it is it's a fancy way of saying it attaches to the receptor. I don't know why we need to learn a new word, but fine, whatever. But SV40 or polyomaviruses attach to receptor via VP1 of our cell. In like all multiplication cycles, you need to attach, penetrate, encode, transcribe, and then replicate and translate, and then bud off. So, of course, the next step is penetration. See, there's this virus here. It's inside a vesicle or an endosome. So here's the endosome, and then it goes through partial encoding. Now, I hope this comes as intuitive, but just in case if it doesn't, then that's okay. I'm here to tell you now. So polyomaviruses need to go to nucleus, not because they need to steal RNA primers like flu does, but because they are double-stranded DNA molecules. So the only way to transcribe the mRNAs for them and to replicate the genome is to start from the nucleus. And that makes a lot of sense because that's where all the cell machinery is. So you have this transport into the cell. You have partial encoding. Basically, you have partial encoding and then you have full encoding. So if you look back at one of my lectures or Dr. Samuel's lectures, you can find out or brush up on the genome of SV40. And it basically has a circular genome. Excuse me, guys, can get some water. I'm still rehydrating myself for my morning run today. Okay, sorry about that. Anyways, you have a circular genome, and the circular genome has an early transcription area and a late transcription area, and it does it through bidirectional transcription, bidirectional regulated transcription. The early proteins are T antigens, which, long story short, they increase the growth of the cell, and growth of the cell is linked with DNA production and protein synthesis, which are perfect, which is perfect for viruses that need to replicate their genome and make proteins. So the T antigens essentially set up the conditions for the replication and transcription translation to occur. Now, continuing on with this picture, so here's the circular genome. Now you have early transcription, like many of the other viruses, and then you have this. Here's your messenger RNA. So because it's a DNA virus, it acts very much the same way as our own human DNA. There is splicing involved. So you have a small genome that's 5,000 base pairs, and yet it codes for so many proteins in a way it does that via splicing. So with the splicing in the early mRNA, you get small T antigen, which drives the cell into the S phase, and you get large T antigen, which large T antigen binds to stuff like p53 to decrease apoptosis, p300 to take control of the cell, specifically take control of cell gene expression, binds to J domain and some other domain which I can't remember, essentially to drive the cell to increase cell growth. So that's the T antigen. So you'll see here that, where's the mouse? So here's the T antigen. The T antigens are then transferred back into the cell, right? Because I said that the large T antigens, I said that the T antigens are needed to set the conditions to drive the cell into growth mode. And when the cell's in growth mode, then it can replicate the genome, make the proteins. So now that the T antigens are in here, you can finally start replicating the DNA of the virus. So all conditions are right. Here it is, it's replicating. And here's a bunch of the progeny genome. So late transcription only is an example of temporal regulation of gene expression, and basically it's you only transcribe and translate the late genes after genome replication is complete. So you can see here, now you make the transcripts of the late genes, and then you bring those mRNAs out. You have splicing, of course, because it's DNA, or it came from DNA. It has to splice out the exons, or the introns, and so forth. Then you have translation of your structural proteins. Okay. Now your DNA is inside the nucleus, and as a general rule of thumb, you can't bring the DNA out of the nucleus because the nuclear pores are too small for DNA to fit through. So polyomaviruses have found a way around this by taking those proteins, the structural proteins, and bring it back inside. Okay, it brings it back inside. Now you have a bunch of the viral DNA plus a bunch of viral structural proteins get virion assembly, and then this naked icosahedral virion capsid buds out of the nucleus, so it steals the nuclear membrane, so it can travel through cytoplasm to the cell membrane, and from here then it has to release back from the intermediate of a enveloped icosahedral virus back into a naked icosahedral virus. And so that's the multiplication cycle of SV40 and polyomaviruses in general.